Hello, BIOME participants. This is Deb Linton. I'm a co-PI on Blue and a faculty member and biology education researcher at Central Michigan University. On behalf of Blue, I want to thank you for taking a look at our slideshow poster on integrating digitized biodiversity data exploration into the undergraduate biology curriculum. If you haven't heard of us yet, Blue is Biodiversity Literacy in Undergraduate Education. We're an NSF-funded research coordination network in undergraduate biology education. Our PI is Anna Monfils, Director of the Central Michigan University Herbarium. Our other co-PIs are Libby Elwood from IDBio and the La Brea Tar Pits, and Lisa White from the University of California Museum of Paleontology. The focus of Blue is preparing undergraduates with the knowledge and skills necessary to work with large and messy data sets containing data about organisms and the abiotic environment. As the volume and accessibility of data about organisms and the environment continues to increase at an accelerating rate, the methods scientists are using to leverage these data to generate and test hypotheses are undergoing a revolution. The availability of these data has increased our capacity to investigate large-scale issues of critical importance such as climate change, resource management, and emerging diseases. Biology programs need to produce biologists with the knowledge and skills to tackle these challenges. The competencies necessary for working with big biodiversity data consist of an intersection of basic data literacy and an added set of knowledge and skills specific to these unique biodiversity data types. Blue is working to develop a community consensus on what those competencies are and to develop strategies and exemplar materials to guide the integration of biodiversity data literacy competencies into introductory undergraduate biology curricula. Of course, the undergraduate biology curriculum is already crowded with content and skills that biology majors need to learn. Our strategy for integrating biodiversity data literacy into this already crowded curriculum is to make sure that the modules are using biodiversity data to address learning objectives that already exist in the curriculum. So we need to look at the content and competency learning objectives for undergraduate biology and identify what subset of those objectives can be addressed using biodiversity data and what biodiversity literacy knowledge and skills students can use to investigate those objectives. 2011's Vision and Change report identified 5 key content areas for undergraduate biology education. Within the content areas of evolution and systems, we believe that there are many learning objectives that could be strongly addressed using biodiversity data. Biodiversity data have a major strength in being able to document change over time in organisms' structures and distributions. Interactions between organisms and between organisms and their environments can also be investigated using biodiversity data, and we have existing modules we've developed to do just that. Vision and Change also identified 6 core competencies or skills needed to be a biologist. This is an area that we believe our modules will be particularly strong. All of our modules require students to apply the process of science and use quantitative reasoning. Biodiversity data also include natural interdisciplinary ties between biology, geology, geography, and other disciplines. And biodiversity data are strongly place-based in a way that can be used to investigate relationships between science and society. So with this strategy in place, we have begun developing curricular materials that meet these criteria. In these modules, students work with biodiversity data to investigate organismal, ecological, and evolutionary concepts using the processes of science. We have over 25 resources posted on CubesHub. All of these resources have been implemented in the classroom, and many have adaptations from other instructors who have used and modified the materials. I'm going to provide an overview of the types of activities you can find in these modules. We have many standalone activities that target specific biological concepts. An example of this type of activity is our amphibian diversity activity. The biology concepts that are being investigated relate to the form and function of osmoregulatory mechanisms in organisms, as well as the influence of the abiotic environment on the distribution of organisms. This is a standalone activity in the sense that it contains step-by-step instructions for students, so the students would not require pre-existing knowledge and skills for working with biodiversity data. These activities are typically at a structured inquiry level, where students are provided with a question and a procedure for investigating the question, but have to complete the procedure and discover the answer for themselves. In this activity, students use IDBio, which many of you may know is an aggregator of digitized natural history collections data. The students search IDBio for records of amphibian species for specific U.S. cities with varying amounts of precipitation. We typically assign each student one city, and students share their data. Students then do a regression with number of amphibian species versus average annual rainfall. On the assessment, students use the regression to predict the number of amphibian species in a city with intermediate rainfall. At CMU, we happen to be living in such a city, so that's a nice connection for our students. The students then go back to iDig Bio to determine the number of species in the collection for that city to see how accurate their prediction was. Here's the data for Mount Pleasant, Michigan. It's actually pretty close to the prediction. Here are some other examples of standalone activities. In addition to a couple of species richness modules, we have activities that use another data aggregator, GBIF, to compare the species ranges of plants and their pollinators, or to look for changes in species ranges over time. We also have activities that use the Arctos dataset to look at the size of collected specimens to see if there are differences in size between specimens collected in different habitats, or changes in specimen size over time. We also have a module that uses the citizen science portal eButterfly to look at shifts in the phenology of plants and their butterfly pollinators over time. All of these activities are structured inquiries with full instructions for students, user guides for the data portals, and instructor materials to help you implement the activity. We also have a sequence of modules designed to be used as a course-based undergraduate research experience, culminating in either an individual or a team research project. This sequence begins by introducing students to the value of biodiversity data and how they can be used using a real-life case study, then introduces the data types and portals, then the ethics of using those data. The next module describes the data standards used by the research community, then we teach students how to download and clean a dataset, and then analyze and visualize data. After students have these skills, they participate in an open inquiry project where they identify their own research question and design and carry out an investigation using digitized biodiversity data. This sequence will be the focus of our working group for BIOME. Of course, instructors could also use a choose-your-own-adventure path and pick and choose from the modules to create your own sequence that fits with your course objectives, or perhaps provide students with sufficient knowledge and skills that you could then use a guided inquiry approach where you pose a specific questions to students and ask them to develop a procedure for answering it using biodiversity data. Blue will be continuing to develop activities in addition to working to develop a community consensus around the biodiversity data literacy competencies. We're also working to develop an assessment that could be used to measure student learning and evaluate the effectiveness of our modules. We'll keep the community informed of our progress. On behalf of Blue, I want to thank you for watching this presentation. If you're interested in getting involved with Blue, please contact me or Anna. We look forward to meeting and working with you.